Hello everyone. In this chapter of Life on Planet Earth, we are going to address the second type of cell division that occur in organisms that reproduce sexually. That second type of cell division is called meiosis. Also, we are going to dive into the genetic basis of sexual reproduction. Now, as we dive into this chapter, I want you to keep in mind the following question. Is there another you in the universe, on Earth? Well, the short answer, it's no. And the question we may ask, it's why? Why is that chance that there is another you on Earth somewhere? It can be in Tuscarawas County, in Stark County, in New York State. It can be in Europe, in Asia, in Africa, in Australia. There is no other you out there across the world. Why? Because of meiosis. Meiosis is the basis of genetic diversity. Meiosis is the reason why, even among your siblings, you may be genetically extremely close, but there is no other sibling that is just like you. That is not possible. Right? The only instance where genetically individual can have the exact same genetic makeup, it's in the case of monozygotic twins. In other words, perfectly identical twins, right? One zygote, right? An egg that got fertilized by a sperm, and by some chance, right? That zygote, after so many divisions, divided into two future organisms, right? So we have that. That's the only instance, right? So the question that we also need to ask is, what is the process by which males produce gametes being reproductive cells, and females produce gametes, again, gametes being reproductive cells? Well, in males, the process by which males produce sperm cells, it's called spermatogenesis, while oogenesis will be the process by which females produce eggs that are going to be fertilized by sperm cells, right? So you understand that spermatogenesis and oogenesis are both type of meiosis, right? We are going to look into the details of meiosis. Now, what is the purpose of meiosis? There are two major purposes of meiosis. One is to actually reduce the number of chromosomes, so that we call meiosis to be a reduction division. In other words, when you consider a human, that human, any cell in your body is going to have 46 chromosomes, right? Now, out of the 46 chromosomes, well, you actually really have 23 that you receive from your mom and 23 that you receive from your dad, right? But this 23 that you receive from mom come from where? Come from the egg, right? That is produced through oogenesis. And the sperm also provided 23 chromosomes, right? That came from dad, right? Now, one may wonder, wait a minute, Dr. Jean, you said that all our cells have 46 chromosomes. These are the body cells, what we call the somatic cells, the cells of the body, your skin cells, your liver cells, but also the precursor of reproductive cells, the precursor of eggs, the precursor of sperm cells. They are all containing 46 6 chromosomes, right? So when a mature sperm cell is produced, a mature egg cell is produced, they only have 23 chromosomes, which means that we are reducing the amount of chromosomes, right, in this gametes or reproductive cells. That's why we say meiosis to be a reduction division. That's one. Two, the second purpose of meiosis is actually to shuffle the genetic information, right? The shuffling of chromosomes and the shuffling of genes. That shuffling of one, the chromosomes, two, the genes, throws in the element of genetic diversity. Now, during the formation of gametes, and again, gametes are sperms and eggs, this is when the shuffling of chromosomes and genes is happening, right? And then once the chromosomes once the eggs and sperm cells are produced, then there is a need for fertilization. That fertilization, there is no prediction whatsoever which sperm is going to fertilize an egg. That's one. Two, on the egg side, there is no prediction whatsoever which egg in the batch of all the eggs is going to be released each month once a lady reaches puberty, right? So there is a random fertilization of eggs by sperms, which adds another actually layer of possibility of diversity of variation, right? It is believed that there is about 70 trillion possibilities for out of all this shuffling of the genetic information, the genes, and fertilization, right? So about 70 trillion possibilities, which means that the chance that you are going to have two exactly identical individuals in this trillions of possibilities is close to zero, right? So each offspring is very unique genetically. The question we want to ask though is, how does meiosis ensures equal distribution of chromosomes to offsprings, which is actually a very important question to ask. So when we look at the genetic basis of inheritance, one can wonder, first of all, what is our genetic makeup? What is the makeup of all our chromosomes? There is a technique that allows us to isolate the chromosomes from cells and actually lay these chromosomes on some kind of map so that we can actually classify the chromosomes based on a number of characteristics. For instance, in the case of the karyotype for a human, now the karyotype, it's actually the map that you see here of the alignment of human chromosomes. That karyotype shows us that the chromosomes are organized by pair. Now remember, we have 46 chromosomes, right? Now, these 46 chromosomes, in fact, there are 23 pairs of chromosomes. One set coming from mom and another set coming from dad. So we have 23 pairs of chromosomes. Now, 23 is the number of chromosomes in a gamete, so that represents the N here. 2 is the pair. right? One you receive from mom, one you receive from dad. So altogether, we have 46 chromosomes. Now, because any cell, such as a skin cell, a liver cell, a muscle cell, a nerve cell, they actually contain twice the number of chromosomes that you would encounter in a sperm cell or in an egg cell. These cells are said to be diploid, while gametes, sperm cells, and egg cells are said to be haploid because they only contain 23 chromosomes. Now, let's go back to the karyotype. When we look at the karyotype, in a human, we are going to classify the karyotype based on the length, the size, and the shape of the chromosomes. The longest chromosomes always come first. Now, when we say this is pair number one, that pair number one has, say, this is the chromosome you receive from mom, right? This would be, the second one on your left side would be the chromosome you receive from your dad, okay? That's pair number one. And then pair number two, there is one from on your left side you receive from your dad, and one on your right side you receive from mom. That's pair number two. Then we have pair number three. Okay? And so pair number three from dad, and this one from mom. And pair number four from dad, from mom, and so on, so that we have 23 pairs. Out of these 23 pairs of homologous chromosomes, you actually have 22 pairs that are rigorously homologous. You have 22 pairs of chromosomes that are rigorously homologous, meaning that for a given set of homologous chromosomes, right? This set of chromosomes, if you consider the genes that are here, say you receive from mom, on the same location, on the chromosome you receive from dad, you are going to find the same gene or an alternate form of the same gene, okay? So that we have 22 pairs of homologous chromosomes that have the exact same genes at the same location for the chromosome they receive from mom and the one they receive from dad. That makes them homologous chromosomes, right? Now, the 22 pairs of homologous chromosomes that are rigorously homologous are said to be autosomes. Autosomes. Now, autosomes are chromosomes in your body that account for anything but sex determination. Okay? They account for everything except for sex determination. That's the 22 first pairs of chromosomes. Then we have the 23rd pair of chromosomes. In the 23rd pair of chromosomes that I'm circling here, in a male, one chromosome is chromosome X, and the other chromosome is chromosome Y, okay? So here we have a chromosome X, and here we have a chromosome Y. That determines a male. And actually, this chromosome X and Y determine the sex of an individual, so that this set of homologous chromosomes, even though they are not rigorously homologous because they don't contain the same, they are not the same size, they don't have the same shape, they don't contain the same genes overall. Therefore, technically they should not be called homologous chromosomes. However, because in terms of function, they account for sex determination, they are both homologous chromosomes. Now, in a female though, female in human, they have two X chromosomes, right? Rather than having one X, females in human have two X chromosomes, while males have X and Y. Now, I was saying that in homologous chromosomes, you are going to have at the same location genes that you receive from dad and you receive from mom, the same genes, right? What we notice though, it's that the version of the gene you receive from mom may be different from the version of the gene you receive from dad, right? That difference account for our differences, right? Some of us are going to exhibit a certain number of features. Say, for instance, your dad has a straight hairline. Your mom has a widow's peak, right? So for the determination of straight hairline and widow's peak is going to be different from mom and from dad. Therefore, even though technically the same gene account for the same feature, however, we see that that feature is different, right? That feature being different, we call the same gene to be alternate forms. So you have a version in mom and a different version in dad. At that point in time, even though these are genes, we actually don't call them genes, we call them alleles, right? So the alleles are alternate forms of the same genes. I want you to remember that. Let me repeat that. Alleles are alternate forms of the same gene, right? Now, where do new alleles come from? Well, new alleles come from mistakes that occur during cellular processes, right? Now, to receive genes from mom, the system in mom has to copy genes, right? The cells have to multiply. Same thing in dad. When cells are multiplying, the genetic information is being copied, right? In the process of copying the genetic information, some mistakes can happen. The kind of mistakes that can actually be transferred to offspring. And when these mistakes are transferred to offsprings, now even the children may not have the same version of the genes as the parents, right? So that overall, you look like your parents. However, you are different from your parents. Why? Because some shuffling of genetic information can happen during the process of meiosis, right? So I want you to remember that new alleles come from recombination of genetic information, mutations, right? And these mutations are not predictable, right? The recombinations are not predictable, right? That is all that account for genetic diversity in a population. Now, when we consider the life cycle in human, now, starting with adult individuals, so we have a male here and a female here. Now, the sex cells, at least the precursor of sex cells, are diploid cells. These diploid cells are going to undergo meiosis. That meiosis for females, again, is called oogenesis, that produces eggs that are haploid containing N chromosomes, in human N being 23, right? In males, meiosis being spermatogenesis produces sperm cells that contain N chromosomes. They are haploid, right? N being 23, right? Now, in the process of fertilization, the egg is going to be fertilized by a sperm cell. And as it gets fertilized by a sperm cell, we have the production of a zygote. That zygote actually reconstitutes the diploid stage, right? So we have n plus n, that makes it 2n, right? 23 plus 23, that makes it 46. Now we have a zygote that is diploid. Now, through the process of development, the zygote is going to start dividing, and that mass of cell is going to progressively start developing into structures that look like organs, such as the limbs, the upper limbs, the lower limbs, the head, and progressively, these features are going to be fine-tuned as the future organism is developing. Eventually, for humans, after 9 months, you have a baby who is perfectly shaped like a human. At that point, you don't have brand new organs that develop. Okay? You don't have brand new organs that develop. So the limbs are going to only grow. The head is only going to grow. You're not going to grow an extra finger. Okay? You're not going to grow an extra leg, which means that after birth, at that time, what happens mainly? It's growth, right? Cells are going to die off and be replaced. So you have some repair that is developing, but you also have some development that is happening, right? What we mean by development is, for instance, a brand new baby doesn't have a beard. If the baby is a male, it doesn't have a beard, right? But in their 20s, that was at one time a baby without a beard is going to appear with a beard, right? Another example, if the baby is a female, that baby obviously has breast, but that baby doesn't have developed breast, right? It's not going to be until the person reaches puberty and beyond that person will start now developing secondary organs, right? That are basically adult organs. Now, once the zygote forms, from then on to adulthood, what is happening in the body is very much a type of cell division that is called mitosis, right? Mitosis occurs after fertilization and all the way until the embryo becomes a fetus and the fetus becomes a baby at birth, right? And from the baby stage to adult stage, all of that, it's mitosis taking place, right? So now let's dive into some overview of meiosis. Meiosis, it's actually 2 consecutive cell divisions. These 2 consecutive cell divisions are characterized by the fact that at the very beginning, before any division, the cell undergo interphase, like any cell would undergo, right? We have the G1 phase, we have the S phase, which is synthesis of DNA, and we do have the G2 phase. And after that, you have cell division, right? You have the division of the nucleus, to be specific, right? The M phase. Now, in this very instance, after the M phase, now we are going to experience another division that occurs without any interphase in between. This is one of the reasons, this is one of the things that is very characteristic to meiosis. That's one thing that I want to mention. The second thing I would like to mention is this. During meiosis, now when we look at the fate of the genetic makeup during meiosis, now we are starting to ask, okay, out of our 46 chromosomes, you have 23 that come from mom and 23 that come from dad, right? Now, if you consider them by pair, right? Pair number 1, right? First division of meiosis. What happens to the chromosome you receive from mom and what happens to the chromosome you receive from dad? Now, that question, we never asked that when we were talking about mitosis, right? That question does not apply, okay? All the chromosomes alike undergo S phase and then prophase, metaphase, anaphase, telophase, and so on. Now we focus on each of the chromosome, right? Out of the 46 chromosomes that the cell started with. That's what we do. But in meiosis, the story is different. We are going to focus on homologous chromosomes so that we're going to take a look at them by pair. And as we take a look at them by pair, we are going to ask the following. Okay, say one we receive from dad and one we receive from mom, what is happening to them, right? Okay, with that being said, I said 2 consecutive cell division, and these 2 consecutive cell division are producing 4 haploid cells. These 4 haploid cells are genetically different. What happens for these cells to be genetically different, we are going to see, right? So we have meiosis 1, which is the first division, and meiosis 2 is the second division. In meiosis 1, what is characteristic? It's that the homologous chromosomes are going to pair up together, okay? Now, as they pair up together, they are going to form a structure that we call synapsis, okay? Synapsis means that the homologous chromosomes are lining up together, and they are forced to line up together because of a set of proteins complex that are going to allow them to come together. I'm not going to bug you with the details, but this set of proteins cause a formation of what we call the synaptonemal complex. The synaptonemal complex. But don't worry about that. I'm not going to bug you with that. What I want you to remember is that the homologous chromosomes are going to pair up. I want you to remember that. Now, as these chromosomes pair up, what happens is that the homologous chromosomes are going to be forced to end up into 2 separate cells, right? And as they end up into 2 separate cells, at the end of meiosis 1, we have 2 daughter cells. In these 2 daughter cells, now for a given homologous chromosome, right, you're going to have one that goes in one cell, the other one that goes in the other cell. At that point, now we say because the chromosomes are now separated in terms of the one that came from mom and the one that came from dad, we say that our cells at that point are now haploid, okay? And again, they are haploid because the set you receive from mom and the set you receive from dad are now in 2 separate cells for a given homologous pair of chromosomes, right? If something is confusing, please let me know. So you can see that here, the chromosomes, are sitting on the equator line so that one is in one hemisphere and the other one is on another hemisphere, which prepares them to end up into 2 separate cells, right? At that point, we have what we call reduction division. The cells are haploid. I want you to remember that. After that, no interphase. The cell is going to undergo a second division. That time, that second division is going to consist of now the sister chromatids are now going to end up toward opposite direction and into 2 separate cells. Okay? We are going to take a look at the details of that. All right. So in meiosis 2, you have the 2 sister chromatids that are going to end up into 2 separate daughter cells, okay? And this is going to lead to the production of 4 daughter cells, because remember that meiosis 1 produces 2 daughter cells. The second division is each of the two daughter cells is going to divide so that we have 4 daughter cells. And each daughter cell is actually haploid. Okay? Now, what you are shown here, it's only the outcome for 2 pairs of chromosomes. Let me go back here. Okay? So you do see, let me change the color here so that you can actually follow along with me. You do see here, this is 1 chromosome and another chromosome of the same pair. And here we have 1 chromosome and another chromosome of the same pair. So we have 2 pairs of chromosomes, right? Okay, so we are going to go back to this. All right. Now, during the formation of the synapses, in other words, during the process where the 2 homologous chromosomes are paired up together, what happens is that the 2 homologous chromosomes, the one you receive from mom and the one you receive from dad, they're going to cross legs. That crossing legs, it's actually called crossing over. Okay? So, in other words, here you have the synaptonemal complex that is going to force the 2 homologous chromosomes to come together. Remember that each of the chromosome has been duplicated during the S phase. Please remember that, okay? And you also do have a prophase like we saw in mitosis, right? Metaphase, anaphase, and telophase. But remember that the S phase occurs, and during the S phase, each chromosome get duplicated, right? In other words, you have this chromosome right here that has 2 legs, so 1 and another 1, okay? So that you have sister chromatids, right? This is 1 leg, this is another leg, okay? And this is 1 leg, this is another leg. Say the red one is the one you receive from your mom and the blue one is the one you receive from your dad. Each of them get duplicated. Now, upon the formation of the synapse, the homologous chromosomes come together and they cross their legs, as you can see here. Notice that the legs are not crossed between 2 red sister chromatids. No. The crossing of legs does not occur between 2 blue sister chromatids. No. Right? The crossing of legs is happening between a blue sister chromatid and a red sister chromatid, so that we have this structure here. And that's what we call crossing over, right? The crossing point is called actually chiasmata, right? Many of them is chiasma, okay? Now, after effectively doing that exchange of genetic information, notice that now these chromosomes are recombined. Now you have one part of the leg that is that of mom and one part of the leg that is that of dad. This is also true right here. Okay, one part from dad, and one part from mom. Okay, so that in the 4 daughter cells that are haploid, you're going to have one cell that is going to have this chromosome that is directly 100% the way it was in dad. This cell is going to have this chromosome that is 100% the way it was in mom. And then we're going to have a cell that has a combination, or a recombination, I should say. So we have this one that is a recombination between dad and mom, and this one between mom and dad, so that we have 1, 2, 3, 4 daughter cells that are genetically different. Now, imagine this happening for 1 pair of chromosomes, right? 2 homologous chromosomes. And now, understand that we do have 23 pairs of chromosomes, right? So what we see for one is going to happen with every single other set of homologous chromosomes out there, right? Now, if you put aside the sex chromosomes and you focus only and exclusively on the 23 autosomal chromosomes, this is basically a lot of possibilities in terms of shuffling of the genetic information. That's a lot of possibilities, which means that chance that two eggs are going to be exactly identical in mom are close to zero. The chance that two sperms are going to be exactly identical in dad are close to zero.